yesterday we finished here when we finally write a system of equation describing the joint changes in maladaptation as a scaled measure of mean phenotype and relative population density relative to the carrying capacity in each patch. And we are looking at so I just draw here the rescaling of all parameters so that we have them in mind. And I redraw the system of differential equations that are going to describe the dynamics of the mean phenotype, or here measured by maladaptation as the distance to the optimal phenotype, and scaled density. And I was explaining to you that because the way those equations are coupled, the equation for maladaptation depends on population size through the ratio of population sizes, and growth of the population depends on mean phenotype through this measure of maladaptation, so through some what is called the evolutionary load, so the fact that the mean phenotype of the population is displaced from the optimum. This affects the population growth rate, and thus the equilibrium population size. So we saw that those interactions, because here the system is positive and here negative, could lead to some kind of a vicious circle of increasing maladaptation in populations that are initially smaller than others. So you can, so what we did when we end up with the system of equation first is just numerically use the system equation to see describe the evolution dynamics of the system. And after a while, you reach some equilibria, and we ended up with different types of equilibria. So in some situation, we ended up with a symmetric equilibrium. So here on this graph is showed the population density as a function of phenotype in pink for one type of host plant and in blue for the other type of host plant. And the dashed line here is the mean fitness in each habitat, and the bell curve, shaded bell curve, represents the density of each phenotype. This is just a schematic representation. So in the first type of equilibrium, what we found is that we had the same density of individuals in both habitats. And if you look at the distance between the optimal phenotype and the mean phenotype in the population, this distance, so the degree of maladaptation, was the same in the two habitats. That's why we say it's a symmetric equilibrium, because there's no built-in asymmetry in the model. Everything, all parameters are completely symmetric in this version. So we can end up as an outcome of joint evolution and demography in a situation where we've got the same density of butterflies on the two type of hosts and the same degree of maladaptation everywhere. So we consider, in this case, the species to be a generalist species because it's exploiting the two host plants in the same way. Doesn't mean that it's perfectly adapted to any, because it can be actually displaced from the optimum in any habitat. But if we would be ecologists and go in the field and count butterflies on different host plants, we'll find the same density, the same use, and so we'll conclude that those hosts are used in the same way by the species. So we will consider the species to be generalist. Note that this species, I mean, even in this case, is generalist in that it uses both host plants, so it's an ecological definition. But if you look in detail, you see that this species is made of populations that are quite differentiated in terms of phenotype and genotypes. So this generalist species is actually made up of a collection of specialized populations in this example. But we could also have, a single peak of phenotype in the middle of this distribution. The other outcome of the simulation that we run were asymmetric at equilibrium. In this case, what we found is that at equilibrium, we had population on one of the host plant where the mean phenotype was not far from the optimal phenotype with very high density, and a very small population on the other host plant with a mean phenotype very far from the local optimum and rather more closer to the optimum in the other habitat. So here we've got a sort sink system where, so yeah, I forgot to say here in this case, because the densities are equal across space, the number of individuals migrating from this host plant to this host plant is just the same. So you've got migration is balanced through space. But here, because the density here is much higher than density here, you've got a net flow of individuals moving from this type of habitat to this other type of habitat, and much less individuals moving in the other direction, just because of the difference in density. So here you've got a sort sink system with a well-adapted population, sending migrants to this sink population where they do not perform well at all because they are really maladapted, and very few individuals coming back. And so in this case, if we would go in the field and observe the situation, we would conclude that the species is a specialist of this pink habitat or host plant, and that it occasionally, a bit by mistake, send out individuals in this other habitat. They're certainly not fit to grow there, and this habitat is clearly outside its fundamental niche because there's no way they can maintain a growing population on this habitat. So, as an exercise, so the idea that you could also try to analyze this analytical model to in which condition you will end up with one type of equilibrium or another. And we can also try to understand what is the effect of migration, selection, genetic variance on the degree of maladaptation and the distribution of population size in any of those equilibria. So in the asymmetric equilibrium, we were not able to find explicit solution to the system of equation that would describe that. And we had to resort to numerical simulation or approximation that work well for very low gene flow, but do not work very well for a higher level of gene flow. But in the case of a symmetric equilibrium, everything is much simpler. So as an exercise, if you want to play with this model, so the idea is that at the symmetric equilibrium, we have N1 at equilibrium equal to N2, and maladaptation in patch 1 equal to maladaptation in patch 2. So what I would like you to prove is that if you look for, so first of all, just to be sure that you know how to do that, how would you find, what would define an equilibrium if you've got a set of equations like that? I can't hear you. Yeah, so what you have to do is to assume that this is 0. So if you do that, and you further assume that there exists a symmetric equilibrium for which this is 0 and this is true as well, you can prove All right, so it's not very difficult. You can do that, probably, if you want to. So let's assume that we prove that. And let's say, now look at what it says actually about the system. So let's try to first interpret this one. So this one is a measure of maladaptation. So it's a measure of scale distance between the mean phenotype and the local optimum. So when will maladaptation be larger in the system? When do you expect a larger distance? Yeah? Yeah. So first result is that maladaptation increase when heterogeneity increases. So if there's a larger distance between the two optima, then you've got more maladaptation in each patch. It's pretty trivial. Then, what is the effect of gamma? What happens if you increase gamma in terms of maladaptation? If this is increased, then the maladaptation is decreased. So what is gamma? Do you remember what it represents? The strength of stabilizing selection. So stronger selection results in less maladaptation. It's also trivial. It's a trivial model. No migration. What happens when you increase migration? Yeah. So maladaptation will always increase when movement rate increases. And the maximal value of maladaptation when the migration rate is really large compared to selection is 1/2. Okay? And it's just also trivial because we rescaled everything. So anyway, you can't really be further from your optimum than the mid-distance between the two optima, okay, in the symmetric equilibrium. Okay, it's not true in this one. But here, if you've got the same degree of maladaptation in both habitats, the maximal distance between your optimal phenotype and your realized optimum is if you sit just in the middle. So migration is always making things worse in this model, and for the generalist. Okay, so now for population size, basically you have part of the same effect, because if you have higher maladaptation, you will have lower population size at equilibrium, with one exception. Which is the effect of the strength of selection now is not monotonous. What happens is that if strength of selection is weak compared to migration, then the population size at equilibrium decreases when the strength of selection increases. And it's just a reverse. If selection is strong compared to migration, then population size will increase when strength of selection increases. Yeah? You had a question? No. So do you understand why? Do you have an intuition about that? So strength of selection will, stronger strength of selection will always result in weaker maladaptation in terms of distance in phenotypic space. But then, if the strength of selection is stronger, a given distance in terms of in phenotypic space to the optimum has more consequences on fitness, okay? If the strength of stabilizing selection is very strong, it means that the Gaussian, the Bell function around the mean phenotype is very picky, and so if you move a little bit away from the optimum, you're gonna have very poor fitness. And so you've got those two antagonistic effects. So if selection is strong, you're better adapted, but any small deviation will have a huge fitness cost. If selection is weak, you will be poorly adapted, but you don't care, because anyway, you do well even if you're poorly adapted. And so the net effect of the strength of selection on population density and demography depends on the relative strength of selection and migration. All right, so that's the equilibrium. Now, another exercise is to find, well, the first one is to find when the symmetric equilibrium is viable. Yeah, because I forgot to say that another solution of that system is that the population size could be 0 in both patches, and this would be also an equilibrium. So how do you do that? So what, yeah, I want a condition on H, on habitat heterogeneity. So how would you do that? Yeah, so here you've got the expression for the population size at equilibrium. And you want to know, so this is not obviously always positive, okay? Because you've got 1 minus another term that could be big. So you want to know when this is equal to 0, this population size at equilibrium. So if you do that, you find that when habitat heterogeneity is greater than a critical heterogeneity, which is equal to 2m plus gamma over m, then you got extinction in both patches. Third exercise. Yeah. No, I'm not sure. You could get extinction anyway. If you got, even if you had slight asymmetry between the two patches, if you had a very, very high migration rate between the two, and very, very, very large distance in phenotypic optima, you could still go extinct, because your emigration rate, basically all your immigrants are deaths. They emigrate from a patch, and they go to another patch where they're completely maladapted, and they die. So if your immigration rate is plus your mortality rate are bigger than your birth rate, you're going to go extinct anyway, even if there's some asymmetry. you have to imagine very high gene flow, very high migration. No, because even if one population, so it's not what makes you go extinct is not the migrants that are coming in. What makes you extinct is a migrant you send out. So if you send out, if the migration rate is ridiculous, like you send all your offspring away, and they're all going to die because they're not adapted to the other environment, then you go extinct. You can get extinct because of too much migration in some heterogeneous environment, so purely for demographical reasons. We'll see if we have time, maybe tomorrow or today, I don't know, that this result of extinction in some heterogeneous habitat is a common outcome of models of adaptation to heterogeneous habitat. So anyway, so there's a critical level of heterogeneity which depends on the migration rate, beyond which you cannot persist in this habitat because you're sending too many of your offspring in some environment where they cannot survive and come back. Yeah, then of course, then the third question is not because you've got an equilibrium that you're going to go to and reach this equilibrium. What do you need this equilibrium to be? You know that? Yeah, it should be stable. The equilibrium should be stable. So the third exercise is find under which conditions the symmetric equilibrium is stable. Do you know how to do that? In general, when you've got a system of equation like that? I guess some of you do, but maybe not the others, so I will explain. So I will explain to you very generally how it works. So when you've got a system, like we have a system of equations, I wrote only two, but we have 4 equations, okay, describing the state variable dynamics in our system of interest. So let's just very generally assume that you've got a list of state variable this. You have n of those. And for each of those, you've got an equation describing the dynamic of x1 as a function of all other variables in the system. All right? This is okay? So now what you do to find an equilibrium of the system is to compute the Jacobian matrix of the system. What is this Jacobian matrix? This Jacobian matrix contains partial derivatives so I will call x bar the set of value of all my variables at equilibrium, of my state variables. So this equilibrium is going to be locally stable when what? When when all eigenvalues have negative real parts. Okay. So we get you all know what an eigenvalue is, basically? Who doesn't know? Okay? Who knows? So eigenvalues are, you can use any program like Mathematica or whatever that was going to compute them for you, or a system, or a matrix like that. So just to remind you, maybe something you've seen in your math course a long time ago. If you have a matrix J like this, and you multiply by some eigenvector, and you get this kind of property. Well, so this is a vector, and this is a scalar. Oh, not necessarily. Then this is an eigenvalue of J. And this is an eigenvector. So basically, you take the matrix, multiply by a vector, you obtain the same vector with every term multiplied by something, the same thing, and then you've got an eigenvalue and an eigenvector. So I will not do it for you. This is an exercise. It's not very hard. And if you do that, you find if I find my page. Yeah. So you find that this condition is that the habitat heterogeneity should be smaller than this quantity. So it depends on the critical habitat heterogeneity where the symmetric equilibrium is not viable. And so basically what it says is that if the migration rate is not too large, which is what we will focus on from now on, then, so here I can call this H max. Then this critical level of habitat heterogeneity is actually smaller than the critical heterogeneity beyond which the symmetric equilibrium is not viable. Which means, in simpler terms, because I think that I'm losing you now, there are values of heterogeneity where the symmetric equilibrium is viable, but not stable. If it's not stable, you're not going to evolve to a state, and instead you will evolve towards this asymmetric state, alternative state that I've described here. So if you plot that, maybe it helps to understand better what's going on. So I've plotted here this critical level of habitat heterogeneity, above which the symmetric equilibrium corresponding to a general species is unstable, and instead you will have an asymmetric equilibrium with a specialist. So you see how this critical habitat heterogeneity changes as a function of the migration rate and for two values of the strength of selection. So what is quite obvious is that in general, when you decrease the strength of selection, and you have got then less difference in fitnesses between the two habitats, the generalist equilibrium is stable for a much larger range of parameters. And the effect of migration is not completely trivial, because so if migration is very small, then the generalist equilibrium is stable. So you have very little migration between the two patches, and you can adapt to both, and you can have two specialists, each adapted to each habitat. Everything is fine. But if you increase, so if habitat heterogeneity is low anyway, you will have the generalist all the time. But if you have a higher, a more heterogeneous environment, if you increase the migration rate, then the generalist equilibrium is not stable anymore, and becomes stable again for very high level of migration. So if I now use numerical studies to see when the specialist equilibrium is stable, because it's not the same condition for the stability of the specialist and the generalist, we found that, so there's this area where the generalist is stable. There's an area where only the specialist is stable. But there's also an area here in parameter space where both equilibria are stable. So it means that depending on initial condition, you can evolve to become a specialist or you can evolve to become a generalist. It just depends on where you started. And this is really interesting. I will show you why. So again, just to illustrate this, in this range of habitat heterogeneity and migration, you expect this kind of situation with two well-adapted population, each specialized on one host plant, but the species is a generalist because it contains those two specialist populations. If you increase the migration rate more, I end up in this area where only the specialist equilibrium is stable, so you have specialization on only one habitat. It could be one or the other, depending on where you started from with a larger population initially. But you cannot maintain adaptation to both habitats at the same time. So you lose completely adaptation on the second habitat. And finally, if you increase migration sufficiently, then you will have, again, a generalist, because you will find equal number of individuals on the two type of habitats, but those two populations will have very little differentiation. So basically, you've got one big generalist genotype that is doing okay in both environments, but you've got almost no local adaptation. So now let's focus on what's going on in this interesting area here. So it's not, arguably, it's not a very big range of parameter space, but maybe it's a relevant, biologically relevant range of parameter space, because those very large migration rates are a bit silly. And see what's going on, what is the consequences of the existence of multiple equilibria there. So for that, I imagine a situation where initially I've got two patches of host plant that are quite separated because around them there's, for instance, a forest. So there's not much movement between those two type of habitat or host plants. Then I imagine that because of, for instance, anthropogenic disturbances or human impact, those two patches become connected. And butterflies can move more easily from one patch of habitat to the other. And then some restoration measures are implemented so that forest grows back between those patches of host plants, and you return to the initial situation where you had very little migration possible between those two patches of host plants. So here I've plotted the function of the movement rate between the two patches of habitat, the total population size. Adding up the number of butterflies found on host patch 1 and 2, And what you can see here is that what we predicted already at the beginning, we've got a symmetric generalist equilibrium. And when you increase the migration rate, population size at equilibrium decreases. This is what we predicted. And what you find as well, but this we did not predict because we didn't do the math, that for the specialist, then at some point you also have another alternative equilibrium, which is a specialist equilibrium, which has a lower population, total population size, because it's exploiting only one patch of host plant and not the other. And there, if you increase migration rate, you also have a decrease in population size. And there's a range of situations where both equilibria are stable. So, but if initially you were there and the movement rate between the two habitats increases, well, you're still declining in size, but at some point, you're not going to decline in size gradually, but you're going to jump this cliff. You're going to actually have a big decrease in population size because suddenly the generalist equilibrium is not stable anymore, and this one is there and can be reached. And so you've got a big drop in population size. And now you've got this conservation measures restoring the natural habitat between those patches. So you decrease the movement rate again, but you not return to this equilibrium because you're stuck in there, in the basin of abstraction of this equilibrium. So here you've got what is called an hysteresis effect. So you've got an effect of history that cannot be reverted on the evolution of your system. So you would have to decrease movement rate much beyond this point so that you jump back to this generalist equilibrium. So this kind of scenario is interesting with respect to the data I show you concerning the butterflies at the beginning of of introducing this model yesterday. Because if you simulate a similar situation where here you've got the population size through time, you have a population at some arbitrary, starting density, then it goes through some symmetric equilibrium, of course, exploiting the two type of host plant, and then suddenly you've got an unusual summer frost event killing all individuals on one host plant, which is what happened in the butterfly population I mentioned. And then because you kill everybody, so of course the population size decreases a lot. But after this disturbance, you do not come back to the system where you had the same density in both patches of habitat. What you end up with is all individuals, so an increase in the density of individuals on one host plant, and a very low density of individuals in the other host plant, which now behaves as a sink, and this one as a source. So you shift from a balanced system of migration to a source-sink system just because of some random disturbance that happened to perturb the system away from this symmetric equilibrium. So yeah, just to show you, well, maybe it will be too long. So this is exactly what the situation that Mike Singer and David Burton described in their population, and I'll come back to that later. So maybe it's not the same mechanism at stake in their example, I must say, because also in their system phenological problems that might prevent recolonization, but at least this model predicts that you could very well see this kind of thing, this kind of very rapid evolution of the niche, because you go from a generalist exploiting two host plant to a drastic reduction in niche width in this system, and very drastic change in demography. So now, there was a lot of questions at the end of my lecture yesterday, which were all very, very good questions. So I prepared a few slides to try to answer those, and I will list them. So the first question that was asked is, okay, but this model assumes constant genetic variance. Seems pretty silly. We know that genetic variance evolves. We discussed that last Friday, that genetic variances would be evolving as any other trait. So what about that? Is it important? And how do you test for that? And how do you improve your model with respect to that? Another important question that was asked was very also to the point, is that all this assume very large population sizes, actually, because you ignore all stochasticity, genetic drift, whatever, and so you're talking about change in population size and everything, but you're not taking into consideration the real stochastic effect of changing population size. Also, I mean, is this assumption of large population size very consistent with the fact that you would have huge effect of migration on those populations? I mean, what is the scale of population size with respect to gene flow? A third question, which is also extremely relevant, is that this model assumes a Gaussian distribution of phenotype and breeding value, while we have migration between very heterogeneous habitat, which we could intuitively think would create deviation compared to this Gaussian distribution. Then somebody asked about what would be the implication of this for niche expansion. And how much those assumptions will matter for it. And finally, but this I won't talk about it, I will talk about it later this week. Movement here is assumed to be completely random and blind. So butterflies are moving around and choosing host plants, basically without little information. And we know that most of the time, dispersal of animals and even plants can be informed by how they perform in their environment and stuff like that. So what would be the effect of having conditional dispersal or informed dispersal? On all this. So I won't talk about this today, but I will talk about the others. So those same questions, I must admit, I asked myself when I did this model during my postdoc, and I actually spent quite a few years trying to answer those after that. So the first thing we did is, because we had this assumption of a Gaussian distribution of breeding value and constant genetic variance, which were the assumptions that worried us the most, already then, we decided to run some simulation, relaxing this assumption to compare with the prediction of our simple toy analytical model. And that's what we did here. We used a model which is not super realistic, I must say, which is called the hypergeometric model, which assumes a large number of loci with only 2 alleles per loci and equal effect across loci, across allelic effect across loci, pre-recombination, and what was also important in this case, we did discrete time simulation, not continuous time, and we had to assume, here we present results assuming selection hacks after recombination, which is quite important. And here you have a comparison of prediction when we vary the migration rate concerning the maladaptation in one patch of habitat, with the model assuming a Gaussian distribution of phenetic value, and this hypergeometric model that does not make this assumption at all. And it works okay. It's not too perfect, but so at least qualitatively, we did not find that relaxing this assumption changed much to our conclusion. Quantitatively, it could make a difference. And if selection happened before recombination, we much less often saw this kind of asymmetric equilibrium, and migrational meltdown. Oh yeah, I didn't even define migrational meltdown. So we call this a migrational meltdown because by increasing migration, you reduce drastically the size of the niche, and you lose completely adaptation to one of the habitats. That's what we call. Yeah. So everything is symmetric in the model there. There's no asymmetry built in. The asymmetry is a result.
No, in the sense, I mean, if there's a difference in the population size, if the population size is alpha, and then we increase migration, would you see?
This is what this was generated by assuming that the carrying capacity of the two habitats is equal. There's no intrinsic differences between habitat quality. What we tried is reverse. We tried to say, okay, but this is silly because there's always differences between habitats. So, well, this works as well if you have, differences in carrying capacity, differences in strength of selection between habitats, and if these kind of built-in asymmetries are not too large, we see the same. But what is interesting here is that you see this, even though, I mean, you had nothing intrinsic in the system creating asymmetry. You get asymmetry because of those feedbacks that I described. And those are completely a bit random. So if you have built-in asymmetries, like for instance, you've got one host plant which is intrinsically better than the other, much more often you will, if you have this asymmetric equilibrium, you will specialize on the better host. But it's possible, I mean, if those differences in qualities are not too large, we could see specialization on the poorest host, just because initially you better adapted to this one and you're locked in the system and you will not evolve to adapt to the better host because of this back between demography and adaptation. This I found interesting because you don't, sometimes you're stuck. That's what this model is saying. It's a model about maladaptation. It says that maybe you have very nice resources that you could adapt to. You have the potential to do it. You have the genetic variance to do it. But because of those feedbacks between demography and adaptation, you're stuck. You're locked in and you do something silly, which is using only one host while you have the capacity to adapt to another. And I like this model because, to me, you understand more about evolution when you understand when evolution fails at producing adaptation. Yeah.
I noticed that your migration rate is completely independent of, say, the degree of maladaptation. So if you had a model where, for example, in the dn by dt, there was a term like m times z1 or zi, that would probably change a lot of features. For example, then the extinction, at least for a perfectly adapted population, the n equal to 0 would no longer be a stable solution, I think. So in general, that would, I think, make a lot of qualitative changes, and a lot of the features may actually go away. Have you?
Okay, I've raised the equation, but the migration is not dependent on maladaptation.
I'm saying if we assume that it's the more maladapted it is, the more it tends to leave its habitat.
No, not what I'm saying. So this would be, if we would, this is perfectly biologically plausible, that if you maladapted, you leave your habitat more often. And this has very different consequences on the evolution of local adaptation, and there are people that more recently have worked on that. But we do not assume that in this model.
I know that, yeah.
Yeah, so is that what you're asking?
That's what I'm asking.
Okay. Yeah. If the migration rate here would depend on Z, that's what you're saying? Yeah, you would have different dynamics, probably. Yeah. So I don't think this was ever implemented in a model with this kind of feedbacks between demography and adaptation, but there have been models trying to look at how this type of informed dispersal would affect divergence or differentiation or local adaptation, or how the phenotype will evolve, but not taking into account the feedback with demography. I don't think I've seen that, but maybe. Okay, maybe, yeah. Yeah, so I've seen that, but the one I saw, maybe I missed. The one I saw, I don't think they had the feedback, but maybe there's some more recent. I don't know. Yeah. No. Well, yeah. Yeah, but somehow this model also, well, okay. So this model, if you talk about demography, so, okay, you could say, anyway, my adaptation is not going to affect my demography. Okay, because I produce enough seeds or offspring, doesn't matter if it's a plant or an animal, that anyway, even if they are really crappy and most of them die, I will produce enough of them so that I will maintain a sustainable population. And most population genetics is built on this model where population size is something independent from evolution. Or you can affect it through drift or things like that, or coalescence, or, but not through the fact that the fitness load or whatever is affecting your demography. So indeed, in this model, I'm looking at a situation where maladaptation has an impact on demography. Okay, so there might be a situation where maladaptation has no impact on demography. But that's not what I'm looking at. Yeah. But I will show you example with, if I have time, about plant migration in the same kind of context. This was inspired by this butterfly story, which I find inspiring. But basically, somehow, it would work better for a plant that disperses seeds or pollen at random in the landscape, rather than butterfly picking host plants and actually using a lot of information about the host plant to choose where they go. The fact is that the thing is that you have a population, you have a growth rate of the population that depends on maladaptation. So if this growth rate is super high anyway, you will get to carrying capacity, whatever. And also, I mean, this life cycle in discrete time, it's also in continuous time. It's also a bit different from what we assume in general of a phase of selection and then a stage of strong density dependence that is putting everybody to the same carrying capacity, whatever the strength of selection before that. Yeah, but at the same time, if this was, I mean, I'm interested in understanding niche limits and range limits. Range and niche limits exist because at some point, plants or trees cannot make it demographically. So I'm interested in what is going on in those set of environmental conditions where what you're talking about is not applying. Because if there's no viable population here, it means that demography is not working there. So if demography is not working, it means that you cannot replace yourself. So, because you don't have the right phenotype in this environment. I'm going to talk about. Yeah. Okay, so we'll talk about that later because I'm going to talk about species range specifically. So I will discuss that. But the model of, so it's true, competition probably plays a very important role. But still, I mean, competition is an ecological factor. You can always wonder, why did you not evolve traits that allowed you to escape competition? So the question, my question is, why did you not evolve traits that allow you to persist in this environment, whatever the constraint in this environment? Is it because you don't have the genetic variation for, to live in there, or is it because you've got other demographic constraints preventing you to evolve despite genetic variance? So this is one hypothesis. There are others, and I will discuss that later. Okay, so about assumptions, because those are also related to those questions. I mean, when do you not adapt to another environment? So basically, at least the kind of simulation we ran when we wrote this paper suggested that those were not that critical, and at least qualitatively, we would see same kind of thing happening even if variance evolved, and even if you had not necessarily constrained the distribution to be Gaussian. But this was not a very thorough exploration, and this model, this hypergeometric model, is only one model for the genetic basis of variation that has also bizarre properties. It might not be super general, and at least there are there are other alternative models for the genetic basis of traits that might create differences. So the conclusion of this work was that you expect a species with a broad niche and a generalist habit to evolve when you've got low habitat heterogeneity, weak selection, either very low or very high migration rates. And you expect a species with a narrow niche to evolve when you've got strong habitat heterogeneity, strong selection, and intermediate movement rates. And those conclusions somehow were reached by others with different assumptions, like assuming a single locus under selection, or in this paper using adaptive dynamics and clonal reproduction. What I found more interesting about this work was this description of this multiple equilibria, which was not really described in the literature before. And that shows that specialization can be, so you can both be a very conserved trait, because you can be locked in this state of maladaptation, and it would be very difficult to evolve a broader niche even if you had the opportunity. And both that it could be a very labile trait, because historical factors and disturbances can create those very rapid shifts in niche breadth when you are in the range of parameters allowing those multiple equilibria to exist. And this predicts that you would expect a lot of geographical variation in host use, and also potentially a lot of temporal variation in host use. And this is exactly what Mike Singer described through its decade-long studies in butterfly diets in California. So this is kind of satisfying, because at least you seem to maybe not understand the detail of that, but understand why broadly it might happen quite often. Yeah. This is my answer. So so this is related to this question of large population size. So one of the answers that you could provide that, okay, 2 patches is a bit silly. How could those 2 patches be big? And I mean, it's not at the scale at which local adaptation would evolve. So what we could imagine more often is a landscape or a metapopulation with many small patches of one host plant and many patches of another host plant. But then it might be a bit different, because you would have to take into account really the spatial structure of those patches, and also competition would be at the scale of patch, but maybe adaptation at a broader scale. And so years after I did this work, Ilkka Ansky was working also on metapopulation, very famous metapopulation of Militia kinkia butterflies in Finland. studied this question of local adaptation. So in his metapopulation, the same species of butterfly was also using 2 different host plants, and there were variation of host plant frequency across the landscape at different grains of spatial variation. And he studied that from an empirical point of view, but also worked on some extension of our model, but taking into account the spatially explicit structure of the landscape with many patches of different host plants, and looking at migration and stuff. And this is a graph from that paper where they looked at the same kind of predictors, like the log migration rate and a measure of habitat heterogeneity. I don't remember precisely how, whether it's exactly the same parameter. It can't be exactly the same parameter, but it has the same kind of purpose, but extracted from a spatially explicit system. And they also found that depending on the value of those 2 broad scale parameters, you could either have adaptation corresponding to unique type of equilibrium in this landscape, and in some other range of parameter, you could have alternative equilibria, so you could actually observe this kind of same shift in the degree of specialization due to change, random change or disturbance or extinction in the system. So if you're interested in that, you could read. Yeah, so what is more complicated in the model is that you've got some special scale of local adaptation. So you can be, for instance, I don't know, you've got a region where you've got lots of host plant 1 and a few patches of host plant 2. So you can have specialists at the level of the patch, or you can be specialized, but at the level of a micro region, because, for instance, you might have here, butterflies that are more specialized in one host plant, and here more specialized in one other host plant. But what they found is that you could also, in this metapopulation system, have an alternative state where you would have, I don't know, a single generalist in the landscape or a collection of different specialists. But there's not only 2 equilibria. That's what I mean. You can have several. You can have specialists, but at different scales. So you can have more than 2 specialists. So concerning population size, some years ago, also with a former PhD student of mine, we looked at a different scenario because so the previous model is predicting that migration is always constraining adaptation. And it does not seem completely right, especially when you are trained in population genetics. Often you heard that, migration is good for inbreeding and so on, that there's an optimal number of migrants to reduce genetic load. And so wanting to see if this prediction that maladaptation is always leading to more maladaptation was not due to the fact that we assume very large population sizes. So we consider a slightly different scenario where we consider this time more like a species that would be distributed along some environmental gradient with different patches. And this could be a species range with large population in the center of the range and smaller population at the margin of the range. And the question was, like, if we play with the same kind of scenario we played it with previously, like we assume that we've got some migration throughout the range. And because of variation in the surrounding matrix, suddenly you start to have much less migration between different patches of habitat. What would be the consequences for, specifically, we focused on the adaptation at the margin of the range. So what would happen for those small marginal populations that are at the margin of the species range? So there was previously a model by Mark Patrick and Gisela Garcia Ramos with the same kind of framework, but using a deterministic model, assuming again a very large population size. And they found that if you disrupt migration, you always end up with increased adaptation in peripheral population. And the reason why is the same as described in the previous model, like gene flow, introducing genes that were selected from the center would oppose local selection at the margins, and there will be asymmetric gene flow because the population in the center are bigger and population on the margin are smaller, so the population of the margin receive more genes from the center than the center receive genes from the margin. But this scenario ignores completely drift and does not consider that there might be other things happening in those small marginal populations. So we run simulations in this case. We have analytical tools to deal with that, and we model some environmental gradient, assuming that you would have some optimal linear some optimal phenotype that will vary linearly through space. That you had some kind of stepping stone population with migration only from one population to the next. And that density will be higher at the center of the range and lower at the bottom. And in this model, density was fixed as a parameter. It was not evolving with maladaptation. It was just classic population genetic model. And what we found is that here you have the mean fitness of the peripheral marginal population as a function of the migration rate throughout the range. And so if you decrease migration, if migration is really large and you decrease it, you indeed find that you have higher fitness in the margins, so you have got increased adaptation for decreased migration for a long time. So if you decrease migration, you've got better adaptation. But at some point, no migration is clearly worse than a little bit of migration. So when you have migration rates that are really small, but maybe order of magnitude that are relevant for many species, and you've got decreased adaptation for decreased migration. So there are several reasons for that. First idea that maybe it's because migration rate in some heterogeneous environment is affecting the level of genetic variance in the peripheral population. And indeed, we find that if you have a little bit of migration rather than no migration, you've got more genetic variance for the adaptive traits contained within each population. So you've got more variance in marginal population if you've got more migrations there. But this is not sufficient to explain what we saw. Because if we just use this value of the genetic variance and feed in the model, the deterministic model of Patrick and Garcia Ramos, we still don't explain why you've got such an increase in the fitness at the periphery when you go from zero migration to a little bit of migration. Yeah. It depends on the demography. There's no demography here, but yeah. It's the same order of, okay, maybe in this case, the load was not very large anyway, okay? But the order, the improvement in fitness when you go from zero to a little bit of migration is the same as the decrease in fitness, or even bigger, but the decrease in fitness when you go from a little bit of migration to a lot of migration. So what I'm saying is that here we discuss about this effect, about whether migration is decreasing adaptation in peripheral populations, but actually this is the same order of magnitude as this effect. So the negative effect is the same order of magnitude as the positive effect of migration. So in this case, I agree that it's just a small effect in this parameter, but it could be bigger. This is just this model, I think. I don't remember which size of population this particular simulation was. So anyway, what we saw is that, I'm going to go through that quickly, is that if you look at the adaptation measured by the distance between mean phenotype and local optimum, the square distance between mean phenotype and local optimum, when you've got a lot of migration, your mean phenotype is displaced from your optimum because you receive a lot of migrant from the central population. So you are not fitting the optimum systematically, but you don't have much fluctuation in the mean phenotype in those populations. When you decrease the migration rate, then your mean phenotype is closer to the optimal value, but it fluctuates around it because of random drift. But if you've got very, very little migration, I mean, on average, you're fluctuating around your optimal phenotype, but you're fluctuating so much because of drift that actually you're, at any point in time, in general, you're very far away from it. What happens is that when you've got no migration at all, you've got a drift selection balance going on in each population, and which means that in the marginal population is small, you've got strong drift, you've got low genetic variance, and selection is completely inefficient. So you fix deleterious mutation in those populations, and they're not locally adapted. And actually, this is confirmed by empirical studies that found that if you look at patterns of local adaptation, you're much less likely to observe any local adaptation if you're looking at small population than if you look at large population. And this is basically the only effect that explains variation in local adaptation is this meta-analysis, is the size of the population. But the population that are next to those marginal populations are bigger, they have weaker drift, higher genetic variance, more efficient selection, so they have a mean phenotype which is closer to their local optimum, but their mean phenotype is actually closer to the optimal phenotype here than the mean phenotype here, because the mean phenotype here has drifted away, because drift is the most powerful force in those populations. If you have a little bit of migration, actually immigration from those larger adjacent populations is reintroducing genotypes that are better adapted to the local condition in the margins. And it restores genetic variance and it also introduces better adapted genotypes. So it's not only an issue of variance, it's an issue of having a population where selection works, and so the genotypes that are in this population are actually better than those ones. And if you've got too much migration, then you've got gene swamping, because those adjacent populations are also poorly adapted because they receive too much gene flow from the central populations that are further away. And so then you introduce migrants that are not well adapted because they are adapted to an environment that is very different from yours. So so this is one study that we did, and then we did other studies, and this is quite important in terms of conservation biology, because a lot of people are really concerned about preserving local adaptation and not mixing things and preventing migration or reintroduction of genotypes from elsewhere. There's also a lot of evidence for genetic rescue when populations are really small and inbred. Actually, they're not locally adapted. So you always do better by introducing individuals from larger populations from elsewhere and reintroducing variation in those populations, because then they will become locally adapted and they basically cannot be worse than what they are when they are isolated. So we did further study trying to look at those conflicting effects of local adaptation and inbreeding, and the conclusion is that the very old prediction that one migrant per generation will be a good strategy is a good strategy, even if you take into account local adaptation. So then the other question is, does it matter to assume that normality, as we always do in those models, just because it allows us to do mathematics? So it's been a number of more recent studies showing that in these environments with heterogeneity, you actually have distributions that are likely to be not Gaussian. And so this is, for instance, one study that we did with Florence Debar, looking at some asexual organism but basically it's the same model as the model I presented to you, except there's no sexual reproduction. And in this case, what you see is that the variance in the phenotypic trait is indeed changing with the migration rate, and also the skew of the distribution is changing, so measured by the third moment of distribution of phenotypic values. And we could show, and others have explored that further, that the response to selection and the level of divergence are actually depending on this skewness, this third moment of the distribution. So basically what I said is not working very well at all when you consider organisms that are asexual. So then another thing, the question about niche expansion and this issue of genetic variance and what you can do with those models. There's another scenario that we worked with a postdoc of mine a few years ago, which is very similar to the one I described to you. So we have two habitats with two different optimal value. But we start initially by we start with a population well adapted to this green environment. And we assume that suddenly another environment is available to be colonized, but the very low density here initially. And so you've got a lot of gene flow initially from there to there, but very little in the other direction. This model was made for plant population. So we assume two stages of dispersal. So you've got seed dispersal, then you've got selection on the seedlings, then you've got density regulation, and then you've got trees or plants producing pollen, and pollen dispersal, fertilizing ovules that will create seeds that will disperse, and so on. So this system is a discrete time model with non-overlapping generations. And we did two things. We had some analytical model with a fixed genetic variance, as we did before. And we ran simulation where the genetic variance was free to evolve, assuming a model where you could draw mutant effect through some kind of Gaussian distribution with some given variance, so that you could have alleles with big effect or small effect. So quite relaxed model of genetic architecture. And so if you use the analytical model, again, you find something similar to what I described to you. So here you've got the population size, this time as a function of the genetic variance, which is taken as a parameter in this model. So depending on the amount of genetic variance you have, you predict different types of equilibrium. And so basically here, I call this niche conservatism, because so you started with a population adapted to the green environment with a large population size in the green environment and very low population size in the red environment. And for this level of genetic variance, which is not zero, which there's potential to evolve, but because of the demographic feedbacks that are described, you do not evolve to colonize the red environment. You stay with your niche, which is adapted to the green environment, and you do not expand your niche. So this is a case of niche conservatism, as I described in the data yesterday. If you reach a critical level of genetic variance, if you go through this critical level of genetic variance, then the other equilibrium becomes stable, and then you've got niche expansion, where you've got an increase in the population size in the second habitat, and then you have a symmetric equilibrium where you as well adapted in both habitats, so you've got higher population size in the two habitats. So this model predicts that there's a critical level of genetic variation that you need for niche expansion. So it's not only that you need genetic variance to adapt to any new environment, you need some critical amount of genetic variance. If you've got a level of genetic variance below that critical level, you will not evolve, you will not succeed in colonizing and adapting to this environment. If you have enough genetic variance to be above this threshold, then you're going to adapt and expand in this new environment. So if you look at simulations, what happens is that often this is a typical pattern. So here you've got, again, the population density. So in the dashed line, it's in the new environment, and in the black in the former well-adapted habitat. So for a very long time, you see almost nothing. So you stay maladapted in the new habitat, almost no increase in population size. And suddenly, at some point, you've got a very rapid shift in population size, and you expand to the new habitat. So you've got not a regular colonization, you have something very flat, and then suddenly a very explosive and rapid growth in this new environment. And this is interesting because in invasion biology, people often see that. They often see a situation where you've got some kind of lag for a long time. The invasive species is not making it, not managing to invade. And at some point, it takes off, and then it becomes completely out of control. And if you look at what's going on at the level of so that's just a demography. So you could imagine that it's just a demographic effect. It's not a demographic effect. It's due to the underlying evolution and adaptation of the population. If you look at what's going on in terms of genetic variance, at the beginning, you've got a low level of genetic variance because you were adapted to only to one habitat. And this level of genetic variance is below the threshold or critical genetic variance that we could define with our analytical model. And at some point, through some kind of random mutation or some random demographic effect, you start to increase this genetic variance. And once you go through this threshold, this is exactly when you adapt and start to expand. So the genetic variance is evolving very slowly, but once it reaches this threshold, then suddenly you have completely different dynamic behavior and demographic and evolutionary behavior. And what we use this model for, oops. Yeah. So we never figure it out completely. Yeah, so but we looked at that. We don't know whether it's, you have to wait until, a mutation with a very big effect arises and start to spread. We don't know whether it's a kind of also feedback with the evolution of the genetic variance, like if you would start a little bit to adapt to the other environment and maintain a higher population size in the other environment, then you start to, have genotypes that are adapted a bit to the two, and then it will increase the genetic variance because migration then will start to have an effect on the genetic variance. We don't know exactly what is causing that. We see that it's very hard in the simulation to identify the mechanism. What we would need probably is to have a model for the evolution of the genetic variance that would allow us to understand why it does that. It could be simply that you wait for the right mutation to occur. But at least, so we don't understand very well the dynamics of the genetic variance, but what we understand is that we can predict what would happen to the system once this level of genetic variance will be reached. Okay. Yeah. Well, no, so when you start to adapt to a new environment, often you see a big increase in genetic variance. Yeah, but the thing is that, so what might happen, it's just an hypothesis. What might happen is that because of the feedbacks that are preventing adaptation, you don't see the spread of those mutations for a long time. And so maybe what you see when the genetic variance is increasing is that just that those mutations can now spread because they escape the negative demographic feedback because they're big enough to escape that. No, there are two habitats. Yeah. I'll finish that and then we'll see the rest tomorrow. So what we can do is we can use an analytical model to predict this critical level of genetic variance needed to adapt as a function of the pollen dispersal probability and the seed dispersal probability. So here you've got this critical genetic variance as a function of pollen dispersal probability for different rates of migration of the seeds. So first thing that we see is that if you increase seed dispersal, so if you go here, it's very low seed dispersal. Here, it's very high seed dispersal. So if you go this way, you always decrease the critical genetic variance needed for adaptation, which means that more seed dispersal in this model is always making niche expansion easier. Okay, so it was not completely obvious at the beginning because seed dispersal has two effects in this context. We already saw that. It puts individuals in the new habitat, so it exposes these individuals to the selection in this new habitat, and if you don't do that, you will never adapt to this habitat anyway. And it adds more individuals to the habitat, so it's increasing the population size in this habitat, which is the first step to adapt to this habitat. At the same time, it's also introducing genes that were selected in the ancestral habitat in this new habitat, so probably it's also preventing adaptation to this new habitat. But the net effect of that is still that, if you increase the dispersal, this demographic effect of adding more individuals to this new environment is making niche expansion easier. If you look now at the effect of pollen dispersal is doing something very different because it's not adding individuals to a new habitat. It has no direct demographic effect. It has only indirect demographic effect because it is introducing genes that were selecting somewhere else and just changing genetic composition of the population without directly adding individuals. And so what happens is that then the effect of pollen dispersal depends on the effect of the amount of seed dispersal. If there's very little seed dispersal, so actually very little individuals in the new habitat, then what you see here is that as pollen dispersal probability increases, then you've got a huge increase in the critical level of genetic variance. So you need more and more genetic diversity to be able to adapt and expand your niche if you have more pollen dispersal. So pollen dispersal is really making it much more difficult to adapt to a new habitat because it's just preventing adaptation by reintroducing always the smarter genes from the ancestral habitat. And basically, it's a main effect of pollen dispersal only for very pretty high seed dispersal. Then you have a more complex effect of pollen dispersal, because if you already have a lot of seed dispersal and already a big population in the new habitat, then pollen dispersal is also acting back to reduce adaptation in the ancestral habitat. And by making the ancestral habitat more poorly adapted, it actually facilitates the colonization and adaptation to the new habitat. So it's a bit indirect, but mostly the effect of pollen dispersal is to slow down niche expansion. And that's what we saw also in the simulation. It was completely consistent with our analytical predictions qualitatively, that if you look at the time you need to adapt to a new environment, if you increase, so those are those isoclines, so here you need 50 generations to adapt to the environment. Here you need a 9,000 generation to adapt to this new environment. And what you can see is that if you increase seed dispersal, you always need less time to adapt to this new environment. But if you increase pollen dispersal, in general, you need more and more time to adapt to this new environment. And it takes so much time that it becomes a bit biologically relevant. If you need 10,000 generation to adapt to this new environment, probably many other things will have changed since then, and you will not make it. Okay, so tomorrow we'll talk about species range. Thank you.